We try to go through around 100 destination plates picked in a day, which translates to about 45 bioassays, depending on the colony counts. The higher the better, obviously. Depending on the species too. Phosmates, the larger scaffolding libraries, usually have smaller amount of colonies that we can use, so they're a little more cumbersome to pick. But yeah, having the 4 machines is nice, and having the XTs are a lot better in terms of the ergonomics, having the stagger system. If there aren't any more questions. Yes, just not today. Basically creating our libraries. Now we're getting ready to prepare those samples for sequencing. And so now we have our deliverable from that area was the 384 well plate, 1 unique colony per well. On this machine, what's going on here is this is called rolling circle amplification. So this is an electron micrograph picture of what the result of what we're going to do here is. So basically we take that 384 well plate, stick it on this plate mate robot, and basically it just takes a microliter of sample, puts it into these 384 well plastic plates. And what we're going to do with these now is basically lice open our E. coli. They've done its job. And now we want to liberate our DNA. And with that DNA, we want to amplify that sample. So what happens is we take this plastic plate, we put it on these pizza oven thermocyclers, heat that up, break open the cell, and add a Templify mixture. Basically incorporates DNTPs, V29 polymerase, and random hexamers. So if I can give a really crappy drawing. So this is basically what's left liberated after the lysis of this E. coli. The Templify has random hexamers, so it'll randomly bind to both our insert and our fragment. And it'll incorporate DNTPs until it finds another random hexamer, and then it'll butt off. When it finds another one, it'll continue to bind. Eventually, over time, you'll have this butterfly effect where you'll have sequencing and then random breaking and then adding of more DNTPs, and that takes place in our incubator overnight. I think it's about 18 hours. And so now we have something that was clear. Now we have something really viscous and thick in that we're hoping that now we have all this amplification taking place.
So when you break open the cells, you're not afraid of the genomic DNA contaminating, or is it going to get screened out later?
That's going to get screened out later.
Okay, so when you do this step, it's just any nucleic acid will get this label.
Right. Okay. Yeah. That cleanup step takes place in the ethanol transfer BET cleanup step.
So that micrograph is genomic DNA mostly?
Yes.
Yeah.
And something else down there.
Yeah, that's in the plasmid. The plasmid.
Yeah.
Yep.
That looks like the artist's signature.
I didn't try.
Instead of E. coli.
Are there any more questions before we continue? Okay, so I'm going to try to proceed a little bit faster. That's my fault. I know you guys want to see the We have that amplification taking place in RCA. We have sequencing chemistry here. Basically what the SciBio machine does is you take your destination plate from RCA, plastic plate that looks like this, place them on this machine, and what happens is the operator will make a forward and reverse cocktail and make a paired end plate basically. You'll have a forward plate and a reverse plate. So you'll have everything that was on the template going in this direction and in the reverse direction so that we can encompass everything. So the primer consists of, the cocktail consists of DDNTPs, DNTPs, our hexamers and our P29. So what happens is now, when it incorporates a DDNTP, a dideoxynucleotide triphosphate, it'll now disassemble that fragment. So now we'll have fragments of various lengths, and we'll thermocycler those plates, and then we'll amplify all of those fragments over and over again. So now we'll have fragments that have blue dyes that are this long and red dyes that are this long. And so that's how the rhodamine dyes will tell us on the sequencers what are being incorporated one base at a time. If there aren't any more questions, then we'll clean up. Put them onto the sequencers. These are ABI 3730s, made in Japan, about $250,000 to $300,000 a piece. When I started here, we ran two platforms, ABI 3730 and Megabase. Megabase has gone obsolete. We went to 454 and Illumina to capture the new sequencers. The plates sit on these bases. Ergonomic injuries in my tenure here, just pulling these off, pulling off the plates. So there's actually a hand tool now, which I don't know where it is. Sorry. One of these. This is what our submitted into Ergo Cup 2008 was. You just kind of put it in there, place it there. You can't really fully appreciate it unless you were to do this about 300 times in your hand for like a year, and then found this bad boy. What can I tell you about the sequence? So what happens is, this array basically will electrokinetically suck up our sample, 96 wells here, suck up the samples like an agarose gel. The smaller fragments will migrate faster through the capillaries here. It's really thin, like hair, until it gets here, where a laser will excite the rhodamine dyes that were attached in our chemistry step that I explained earlier. That rhodamine dye will emit a wavelength. The base calling software of this machine will say with 95% certainty that the base that flowed through here and gave out this wavelength had a blue rhodamine dye, which translates to a guanine, and then the map will be sequenced on here. Of course, none of these are on. Yeah. So, if you can imagine, this room once was filled with ABI 3730s. And Megabase was in the other room. We had something like 75 running machines. What those machines could do in one day at full capacity, the 454 sequencer can do in one 8-hour run. So, in terms of the cost, obviously more expensive for the reagents here, but in terms of the read lengths being a lot higher, we still have 3730s for that purpose. And people in Brazil and people in Florida that like their citrus products genome sequenced, like Sanger being sequenced on it instead of 454. It's a little bit cleaner, obviously, so that's why we're still running them. 454 has its limitations as well, obviously shorter reads. Operator costs are a little bit more expensive, reagent costs are a little bit less, but turnover is a lot higher. Yeah.
Should I keep this one?
For a time. I think we're going to sequence cotton and citrus on here, and then eventually ABI will be gone from us.
You gonna sell them? Or?
Actually, we've been giving them to academia. My girlfriend works at University of Washington as a genome scientist. We gave her 2 of these machines, so I don't know. If you're nice to me, maybe I'll give you one. But yeah.
I work with you.
Okay. But yeah, the contracts are really expensive to keep with ABI.